Every year, billions of birds travel thousands of kilometers with astonishing accuracy. Plants convert sunlight into chemical energy with an efficiency that still inspires engineers. And inside your own body, enzymes carry out chemical reactions millions of times every second. At first, these seem like completely different biological processes. But over the last few decades, scientists have begun asking a surprising question. Could they all share something in common? Not DNA, not evolution, but quantum mechanics. This is the emerging field of quantum biology. And unlike many popular myths about quantum physics, it is built on experiments that can be tested. What does quantum have to do with life? Quantum simply means the rules for very small things: electrons, atoms, and light. Most living cells are warm and busy, which usually hides fragile quantum effects. So we'll ask a practical question. Where do quantum rules give biology a small but useful edge? We'll follow 3 clear stories: bird navigation, photosynthesis, and fast chemical reactions, and then show how scientists test those claims. Before the case studies, 2 ideas help: superposition and tunneling. Superposition means a tiny system can hold several options at once until something forces a choice. Tunneling lets a particle cross a thin barrier it cannot climb. The thinner the barrier, the likelier the crossing. We'll meet both at nanometer scales and femtosecond to microsecond times in proteins and pigments. Case study. 1. How some birds sense Earth's magnetic field. Observations of bird migrants keeping direction on cloudy nights raise the question, how? And the answers are hidden in the retina. The retina contains a protein called cryptochrome. Cryptochromes are a class of ancient, flavin-containing photoreceptor proteins that regulate blue and ultraviolet A light responses across plants, animals, and fungi. They are critical for setting daily internal biological clocks, mediating plant growth, and even helping certain animals sense the Earth's magnetic field for navigation. Cryptochrome has a small light-harvesting cofactor called Fad, which absorbs blue light. Blue light moves an electron along a short chain. 2 molecules each hold 1 unpaired electron, a radical pair. Each unpaired electron has spin, a tiny magnetic property. The pair flips between 2 combined spin states. Earth's weak field slightly shifts how often those states swap, a tiny energy change. That small bias can change the chemical outcome a little. Spin correlation plus field-biased mixing is enough to matter. The result may be a faint orientation signal the visual system can use, not a glowing arrow, but a subtle bias. Tests make it concrete. In a Helmholtz coil arena, paired coils that create a uniform, rotatable field, birds' preferred headings rotate when the field rotates. When the field is rotated, the headings rotate too. Experimental controls in such scientific endeavors are highly important. Darkness to remove star cues, sham coils that look active but add no field, and removal of landmarks. Headings must track the field, not the room. When very weak radio frequency fields, tuned to the spin transition frequencies, are added, orientation degrades, right at resonance. When experiments move off those special frequencies, the birds recover their headings. In short, when spins are nudged, the behavior fades. When the nudge is removed or the frequency is shifted, it returns. That on-off signature matches a radical pair magnetoreceptor. Takeaway: Blue light starts a cryptochrome reaction. An electron transfers. A radical pair forms. Spin switch between singlet and triplet. Earth's field gently biases the mixing. Behavior shifts in predictable, testable ways. Open questions remain: protein variants, lifetimes, and wiring, but the current evidence already guides careful experiments. Quiet physics, guiding flight. Humble, testable signals, small, steady, and useful. How do we test it? In a Helmholtz coil arena, paired coils create a uniform magnetic field that we can rotate precisely. When the field rotates, the bird's preferred headings rotate with it, under darkness and sham coil controls. When weak radio frequency fields matching spin transition frequencies are included, orientation can be probed. Orientation degrades on resonance and recovers off resonance. This on-off pattern is the fingerprint of a radical pair sensor, when controls are strong. Case study 2. Photosynthesis moves light's energy with high efficiency at room temperature. Interest grew because simple models could not explain such reliable transfer. Where it happens, inside chloroplasts, on stacks called thylakoids, in light-harvesting complexes that pass energy to a reaction center. One photon excites a pigment. An electron moves, leaving a hole. Together they act as an exciton, an energy packet that can hop between pigments. How do we see that guidance? With ultra-fast snapshots. Laser flashes separated by femtoseconds. The data look like panels of colored spots. Linked spots show coupled sites. Tiny beats show short-lived coherence. These patterns shift with temperature and with protein layout, evidence that structure tunes the flow. Protein design sets spacing and which tiny vibrations are allowed. Too rigid, energy can get stuck. Too noisy, guidance washes out. In the sweet spot, moderate random jostling helps steer around traps while coherence lasts long enough to help. A little well-timed noise helps. Small jitters nudge the exciton out of dead ends and keep it moving. Randomness used on purpose. After a few trillionths of a second, coherence fades. Ordinary vibrations carry the energy into the reaction center. Result: near-perfect delivery at room temperature, engineered by structure, not magic. One photon in, charge out. Brief coherence finds good routes. Vibrations finish the handoff. High efficiency, by design. For a brief moment, the exciton explores several routes at once. That short coherence boosts easy paths and lets harder ones fade. Ultra-fast laser experiments take snapshots a few femtoseconds apart. Linked spots reveal coupled sites. Small rhythmic beats mark short-lived coherence. Protein design matters. Distances and allowed vibrations set a sweet spot. Not too rigid, not too noisy. Coherence sets up fast options. Ordinary vibrations finish the transfer into the reaction center. Charge separated. Case study 3. Some enzymes use quantum tunneling to move protons efficiently. Interest rose when measured rates were faster than simple over-the-barrier models predicted. In this picture, a donor and an acceptor are separated by a thin barrier. Over the top is slow. Through the barrier, when it's thin or short, tunneling can be faster. As the barrier thickens or widens, the tunneling chance drops steeply. Evidence 1. Isotope substitution. When hydrogen is replaced with heavier deuterium, tunneling weakens, rates drop, and temperature slopes remain shallow compared with a plain Arrhenius curve. This kinetic isotope effect is a classic tunneling fingerprint. Evidence 2. Protein gating. Tiny, hinge-like motions briefly shorten donor-acceptor distance and align bonds, thinning the barrier in real time. When the alignment window opens, tunneling probability spikes and the transfer fires. Put together, heavier isotopes slow rates without steep temperature dependence, while micromotions open brief low-barrier paths. These paired signatures are hard to mimic with purely classical hops. Mechanism in one sentence, protein structure sets who can react. Thermal jostling and brief coherence thin the barrier, just long enough for a proton to tunnel. Ordinary vibrations carry the rest. Timing and distance matter most. Sub-angstrom changes can swing tunneling rates by orders of magnitude, and hydration tunes both flexibility and alignment windows. The rule is design, not miracle. Biology shapes geometry, barriers and noise so useful quantum effects appear briefly, do work, and fade. Fragile physics harnessed by robust dynamics. Summary: tunneling at room temperature shows clear fingerprints, an isotope effect, a shallow temperature slope, and distance gating. These guide targeted, pre-registered tests in specific enzymes. Vision begins with a rapid event in rhodopsin. The retinal molecule flips shape, from 11 cis to all trans, in about 200 femtoseconds. A brief quantum push helps start the first bond twist. Decoherence follows quickly, and vibrations carry the rest. Amplification in the visual pathway turns this tiny change into a reliable signal. Strong claims need strong tests. Behind every claim sits careful preparation, pre-registered plans, blinded labels, and calibrated fields, so results speak for themselves. For magnetoreception, we watch headings follow a rotated, calibrated field. When controls hold, confidence grows. Not a single frequency, but full sweeps, with uncertainties, reveal the shape of the effect. In photosynthesis, side-by-side designs and temperatures show how structure quietly steers energy. In enzymes, swapping isotopes and tiny distance tweaks expose tunneling's footprint without drama. Patterns that resist a plain Arrhenius curve hint at a thinner, faster path. The point isn't a checklist, it's a map, from prediction to measurement, so anyone can retrace the route. Quantum biology asks where living systems exploit quantum effects in specific, testable situations, and where they do not. With each replication, the picture sharpens, and the next questions come into focus.